Welcome to the Deep Dive, where we unpack complex topics and distill them into essential, engaging insights just for you. Today, we're diving into a truly fascinating corner of the biological world, one that often gets mistaken for something else entirely: the Oomycota.
That's right. When you hear about devastating plant diseases or maybe some strange aquatic organisms, your mind probably jumps straight to fungi.
Yeah, mine too, usually.
But as we'll uncover today, the Oomycota have their own unique identity. They've got a surprising evolutionary story and, honestly, a profound, often overlooked influence on everything. Think agricultural history right through to the health of our planet's ecosystems.
And we're drawing our insights today from a really detailed chapter in Introduction to Fungi, 3rd edition, by John Webster and Roland W.S. Weber. Our mission for this deep dive is, well, to clearly explain the intricate science of Oomycota. We want to focus on what makes them distinct, how they navigate their world, and their significant real-world roles. And we're doing this all without a single visual.
Just vivid descriptions and clear explanations. Hopefully you'll have a few aha moments along the way.
Exactly. So let's start with that central mystery: the Oomycota identity crisis. What are these organisms, and why do they so often get lumped in with true fungi?
It's a great question because, superficially, they behave very much like fungi. They grow in similar ways, these thread-like structures. They secrete enzymes to break down food. They form spreading networks. Historically, they were actually classified as fungi.
Right, okay.
But despite that resemblance and, similar ecological roles, Oomycota is sometimes called Peronosporomycetes. They're a distinct evolutionary branch. They actually belong to the kingdom Straminipila. That's a group that includes things like diatoms and brown algae.
Oh, wow. So quite different then.
Very different. Think of it like this. A dolphin looks and acts a lot like a fish,
Yeah.
right? But it's actually a mammal. The Oomycota are kind of the microbial equivalent. Similar lifestyle, totally different lineage.
Okay, so if they look and act like fungi, what are the core biological differences that really set them apart? What should we, really pay attention to here?
Right. That's where we need to dive into their cells. There are several fundamental distinctions. First off, their main body threads, called hyphae, are typically coenocytic.
Coenocytic, what does that mean exactly?
It means they're like long, continuous tubes. They generally lack internal dividing walls or septa. Most true fungi have those partitions, kind of like rooms in a house. Oomycete hyphae are more like one long open corridor for all their cellular contents.
Huh. And does that open structure give them some kind of advantage, like for growth or moving nutrients around?
It certainly can. It allows for really rapid cytoplasmic streaming, which, can be beneficial for quick growth and nutrient distribution, especially when they're colonizing a new food source. Now, another major difference, and this is a big one, all their vegetative structures are diploid.
Diploid, okay, like our cells. 2 sets of chromosomes.
Exactly, just like most of our own cells. But in stark contrast, the main body, the vegetative part of true fungi, is usually haploid, only one set of chromosomes.
Yeah.
This fundamental genetic difference, it really shapes their entire life cycle.
And it doesn't stop there, right? I remember reading that even their cell walls, the very structure holding them together, are different.
Exactly. This is another huge one. True fungi, their cell walls are made primarily of chitin, the same stuff in insect exoskeletons.
Right.
But Oomycota cell walls, they're composed mainly of cellulose.
Cellulose, like plants.
Precisely, which is the primary component of plant cell walls. It's a really striking parallel to the plant kingdom, not the fungal one at all.
And what's truly surprising, I thought, was that even their internal machinery, like the mitochondria, those powerhouses, show this divergence.
You've hit on another crucial distinction. Yeah, their mitochondria have these internal folds, the cristae, that are tubular, like little pipes, rather than the flat, sheet-like ones, the lamellate ones found in true fungi. Again, it's another subtle but significant parallel with plants. Wow. And if that wasn't enough, their primary way of storing carbohydrates isn't glycogen, like fungi use. It's a unique polymer called mycolaminarin. And this often collects in these distinctive internal structures called fingerprint vacuoles.
Fingerprint vacuoles, okay. So if we pull all these details together, the cellulose walls, the diploid structures, different mitochondria, unique storage compounds, what's the big picture takeaway here?
The key insight, really, is convergent evolution. Oomycota and true fungi have arrived at very similar lifestyles and ecological roles, particularly as decomposers or pathogens. But, and this is the crucial part, they've done so via entirely different evolutionary paths.
So they look alike, do similar jobs, but their basic biology shows they're, distant cousins, not siblings.
Exactly. It's a powerful reminder that life finds many ways to solve the same problems.
Speaking of solutions, let's talk about how these organisms move and spread, because one of the most defining and, frankly, incredible characteristics of Oomycota is their modal asexual spores, called zoospores. These aren't your typical airborne fungal spores we might think of, are they?
No, not at all. These zoospores are, well, they're kind of their secret weapon, especially for life in water or wet soil. They're heterokont.
Heterokont.
Yeah, I mean, they have two distinct flagella, or tails, that are very different from each other. You can think of like a tiny high-performance boat. One flagellum is long and feathery. It looks almost hairy under a microscope, and it acts like a powerful outboard motor propelling it forward.
Yep.
The other one is shorter and smooth, the whiplash type, functioning more like a steering rudder. This combination makes them incredibly agile and really efficient swimmers.
And it gets even more sophisticated because some Oomycota use this kind of two-stage dispersal strategy, which sounds pretty unique.
Yep.
Tell us about deplanetism.
Ah, yes, deplanetism. It's a brilliant adaptation, really. Some Oomycota display this, meaning they produce two distinct types of zoospores, one after the other. The first one, called an auxiliary or primary zoospore, is often shaped like a grapeseed, and it has both flagella right at its very tip. It seems designed for quick, initial dispersal, and it often basically forms a protective wall around itself quite rapidly.
It insists, so it stops swimming.
Temporarily, yes. But here's the clever part. From that auxiliary cyst emerges the second type, the principal, or secondary, zoospore. Now, this one is different. It's kidney-shaped, and crucially, it has its flagella inserted laterally on the side in a groove.
And how does that sideways insertion make a difference?
It completely changes its hydrodynamics.
Yeah.
It gives it a massive advantage. With one flagellum pointing forward and the other sort of trailing backward, it achieves a much more efficient propulsion. These principal zoospores can swim up to 3 times faster and with much more directed movement.
Wow, 3 times faster.
Yeah. It allows them to really vigorously explore their environment, actively seeking out suitable surfaces or, importantly, hosts. Once they find one, they quickly insist again, shedding their flagella and secreting a new cell wall, ready to germinate and start growing hyphae.
That's a remarkable strategy for finding a foothold, and I read, for some, it doesn't even stop there.
Indeed. Species like Saprolania, a common water mold, exhibit polyplanetism.
Polyplanetism, more stages.
Exactly. It means a principal cyst can actually release another principal zoospore before it finally settles down and germinates. Imagine the dispersal power this gives them. Each release is another opportunity to find new territory or hosts. It makes them incredibly effective colonizers, especially in moist environments. This multi-stage motility is really a key insight into their success, particularly as pathogens.
Okay, so beyond these amazing modal spores, Oomycota have a surprisingly sophisticated sexual reproduction system. And again, this shows their distinct biology and apparently led to some groundbreaking discoveries.
Absolutely. Remember, they're diploid in their vegetative state. So meiosis, that process of having chromosomes for sexual reproduction, happens within specialized reproductive organs called gametangia. This is different from many true fungi, where it often happens right after nuclei fuse. And their sexual reproduction is oogamous.
Oogamous.
Yeah, it's a term that just describes having distinctly different male and female gametangia, different in size and shape. You typically have a large, often spherical, female oogogonium, and then a smaller male antheridium.
Okay, so what does this highly differentiated sexual system mean for understanding them? You mentioned discoveries.
Right. Well, this structure led to the incredible discovery of hormonal control in their reproduction. This was particularly worked out in the genus Achlea back in the mid-20th century by scientists like John Raper. It's like a chemical conversation.
A chemical conversation? How does that work?
It's truly one of the most fascinating aspects. In what are called heterophilic species of Achlea, meaning you need 2 different strains, sort of like male and female. For sex, it's a precise chemical ballet. A female strain will secrete a powerful steroid hormone called antheridiol. And this stuff is active at astonishingly low concentrations.
Wow.
This hormone acts as a signal, basically telling the male strain, hey, I'm over here, and triggering it to grow its antheridiol branches towards the female. Then the male strain responds by secreting another steroid hormone called ooganiol.
So it talks back chemically.
Exactly. And ooganiol stimulates the female strain to form those ooganiol initials, the beginnings of the female structures. It's like a microscopic chemical dating app, seriously. Each partner sends and receives specific hormonal signals to coordinate the whole process, even directing the growth of their reproductive bits towards each other.
That's astounding. Such complex interactions in seemingly simple organisms. And I read something about relative sexuality, too.
Yeah, that's another layer of complexity. It means a strain isn't rigidly male or female. Depending on the hormonal strength or the response level of its partner, a particular strain might act as the male in one pairing, but as the female in another.
Really? So it's flexible.
Very flexible. It just highlights their remarkable adaptability and how intricate these hormonal systems are. They don't just kick off reproduction, they guide growth. They even stimulate enzymes to soften cell walls to allow fertilization. It's a powerful example of precise biological communication happening at the microbial level. Now, shifting gears a bit, these seemingly simple organisms, the Oomycota, especially certain groups, they've had an immense and sometimes, frankly, devastating impact on human history. And they continue to shape our world today. They're incredibly widespread. You find them thriving in diverse environments, freshwater, marine ecosystems, soil, and of course, crucially on plants. While many are saprotrophic, nature's recyclers breaking down dead stuff, a significant number are really powerful parasites.
And here's where their impact really, really comes into focus for most people, right? I mean, think about the Great Irish Potato Famine back in 1845 to 1848.
Exactly.
That's devastating event, mass starvation, emigration. It altered global demographics, politics. It was directly caused by an oomacetes.
Phytophthora infestans, the pathogen behind potato lake blight.
A single Oomycete species with world-changing consequences. And their influence extends beyond just causing famine. It was the downy mildew of grapes, caused by another Oomycete, Plasmopara viticula, that actually spurred the research leading to the development of the very first fungicide Bordeaux mixture.
Wow, I didn't know that. So they didn't just cause disease, they actually pushed us to invent ways to fight plant diseases.
Precisely. They literally changed how we approach crop protection. And these aren't just historical footnotes. Oomycota remain major plant pathogens today, responsible for billions, literally billions of dollars in crop losses worldwide every year.
Billions. And it's not just plants, is it? No, not at all. Species like Sapropelma, those water molds we mentioned, cause serious infections in farmed fish, especially salmon. That leads to significant losses in aquaculture. And then there's Aphanomyces astacei, infamous for causing the devastating crayfish plague in Europe, which severely impacted aquatic biodiversity.
It sounds mostly like bad news, but you mentioned earlier, it's not all doom and gloom. Some might be useful.
That's right. It's fascinating, actually. Some species, like Laginidium giganteum, are being seriously investigated for their potential in biological control, specifically as parasites of mosquito larvae.
Controlling mosquitoes, that would be huge.
It's a very promising area of research, yes. So, you see, they play truly diverse roles in ecosystems. They're decomposers, they're destructive disease agents, but they might also be potential allies for us in managing pests or other issues.
Okay, let's maybe delve a little deeper into some of these key groups and their strategies, starting back with the water dwellers.
Sure. The Saproliniales, often just called water molds, they're really abundant in aquatic environments. Genera like Saprolinea and Achia are the common examples you'll find. They're mostly saprotrophs, cleaning up plant and animal debris in ponds and streams. But, as we noted, they also include those significant fish pathogens and their unique multi-stage zoospore life cycles, that deplanetism and polyplanetism we talked about, that's really key to how they spread so fast in water.
Okay, then moving more onto land, you mentioned the Pithiales.
Right, the Pithiales. This group contains two exceptionally important genera, Pythium and Phytophthora. Pythium species are, well, they're everywhere in soil, often just acting as decomposers, breaking stuff down.
But they can be trouble, too.
Oh, yes. They can be opportunistic plant pathogens, causing damping off diseases, especially in seedlings. It's where they essentially rot the young plant right at the soil line. And some Pythium are even mycoparasitic.
Mycoparasitic. They attack fungi.
Exactly. They attack other fungi. An interesting ecological twist there.
And Phytophthora. That's a name that, as you said, strikes fear into farmers and gardeners.
It certainly does. Phytophthora literally means plant destroyer, and it lives up to its name. This genus includes the infamous Phytophthora infestans, the potato blight pathogen we discussed. But there are many others causing devastating blights, root rots, stem rots, sudden oak death. Just a huge range of diseases on an incredibly wide array of plants.
What's fascinating is how these pathogens adapt for such efficient infection. How do they do it? We know their zoospores are good swimmers, but what happens when they actually reach a host plant?
Right, the infection process itself is highly adapted. For example, Pythium zoospores, They're known to use chemotaxis. They can actually sense chemical signals released by plant roots and actively swim towards them, essentially aiming for their target.
Wow, targeted swimming.
Yeah, and once they reach that root surface, they can insist and penetrate the host tissue in, well, sometimes as little as 30 minutes. It's incredibly fast. And many phytophthora species, once they get inside the host cells, they form these specialized structures called haustoria.
Haustoria, like little probes.
Exactly like probes. They're tiny, nutrient-absorbing structures that penetrate the host cell membrane without actually breaking it. This allows them to sit inside the living cell and just siphon nutrients directly from the plant. It's a very intimate and efficient parasitic relationship.
Okay, then there's another major group, the Peronosporales, the downy mildews. How do they fit in?
The Peronosporales, yes. They are another critical group, especially in agriculture. Now, unlike many of the Pithiales, these guys are mostly obligate biotrophs.
Obligate biotrophs,
No.
meaning they have to feed on living tissue. They can't survive on dead stuff.
Precisely. They can only grow and reproduce on living host tissue. And they tend to be highly specialized, often having a narrow host range, maybe infecting only a few related plant genera. Now, for their terrestrial lifestyle, a really key adaptation is that their sporangia, the structures that produce spores, often germinate directly by putting out a germ tube instead of releasing those swimming zoospores.
Ah, so they've kind of ditched the swimming stage for life on land.
In many cases, yes. This allows the whole sporangium to act like a functional conidium, basically a dry, airborne spore, which allows for really effective wind dispersal, a major shift from their aquatic ancestors.
That makes sense. So they blow around in the wind instead of swimming.
Exactly. This group includes genera like Peronospora, which causes diseases like blue mold of tobacco, and Plasmopara, which, as we mentioned, includes the grapevine downy mildew that famously led to the discovery of Bordeaux mixture. And their interactions with host plants have been absolutely pivotal for our understanding of plant resistance mechanisms. Things like the gene-for-gene relationship.
Cure for gene.
Yeah, is where a specific resistance gene in the plant recognizes and counters a specific virulence gene in the pathogen, like a locking key, and also systemic acquired resistance, or SAR, which is a broader, long-lasting defense response in the plant. A lot of this fundamental work was done studying these downy mildews, often using model plants like Arabidopsis thaliana.
So these pathogens actually helped us understand plant immunity?
It really did. They pushed
Yeah,
the
cool.
boundaries of that field.
And finally, you mentioned albugo and the sclerosporiaceae.
Right, just briefly. Albugo causes those distinct white blisters or white rusts you might see on plants like shepherd's purse or radishes, members of the crucifer family. And the sclerosporiaceae, those are the downy mildews specifically of grasses and cereals. They're particularly impactful in tropical regions, causing significant crop losses there. These groups just show further variations on the themes, different ways of producing spores, like albugo's sporangia forming in chains, and unique host interactions. So after this deep dive, what does this all really mean for you listening? Well, the Oomycota, while they're often mistaken for fungi, are clearly a distinct and ancient lineage with truly profound impacts on our world. Their unique biology, everything from their cellulose walls and their diploid vegetative hyphae to those incredibly motile zoospores and complex hormonal signaling, it all really sets them apart.
Yeah, and we've seen how their specialized life cycles have made them formidable pathogens, whether it's the efficiency of those fast-swimming principal zoospores or the intricate chemical dance of their sexual hormones or even their adaptations for wind dispersal. These strategies allow them to cause epidemics that have, as you said, literally shaped history and continue to challenge modern agriculture.
Absolutely. From the devastating Irish potato famine right up to today's sophisticated crop protection strategies, which involve targeted fungicides and breeding-resistant plant varieties, understanding these fungi-like organisms isn't just some academic exercise. It's genuinely crucial for global food security, for ecological balance, and even for things like public health when you consider their impact on aquaculture and their potential in biological control.
They truly highlight the immense, often unseen diversity of life out there and the incredibly intricate ways organisms interact with their environment and with each other. Often, as we've seen, with surprising and really far-reaching consequences. This deep dive into the Oomycota has certainly given me, and hopefully you, a lot to think about. It really makes you wonder, doesn't it? If similar lifestyles, like being a pathogen or a decomposer, can evolve independently multiple times in completely different lineages, what does that tell us about the fundamental pressures shaping life on Earth?
It definitely raises a big question. What other hidden evolutionary convergences like this are out there, waiting to be discovered? Discoveries that might fundamentally change how we classify life, how we understand life's intricate web.
Exactly. Well, thank you for joining us on this deep dive into the fascinating world of Oomycota. We hope you're now feeling well informed and maybe just a little more curious about the hidden microbial world all around us. Until next time, keep exploring.